What if I say that life on Earth depends on our sun, plant, and the cyanobacteria? Would you believe me? Let me tell you why. Plants have this special ability to utilize the sunlight and carbon dioxide they receive in order to create their own food. And the byproduct of this process is what keeps the life on Earth going, which is the oxygen gas. In addition, animals like cows, goats, etc., primarily consume plants, and we people depend on our livestock as a food. Adding to that fact is that plants can also be consumed, and some are even used as a herbal medicine. While on the other hand, plants uses our carbon dioxides to fuel their process of making food. So we could say that it is a mutual relationship. But how does plant utilize light to make their food? The quick answer would be the chemical reactions of carbon dioxide with water driven by the light in order to yield carbohydrates and oxygen gas. These carbohydrates will be broken down in order to create energy that will be discussed in deep later. So, would you believe me now that life depends on them? Now that you're already convinced, let's talk about what makes plants special and why photosynthesis plays a vital role in life on Earth's system. Plants in general consist of this organelle known as the chloroplast. It is the site where the photosynthesis occurs for the eukaryotes. Shown on the left-hand side of the figure is an actual electron micrograph of chloroplast. And on the right-hand side is the schematic diagram of chloroplast with its part highlighted. Now let's move forward and deepen our view with the chloroplast by discussing the significant components starting with the stroma. The stroma is enclosed in the inner membrane of the chloroplast. It is a concentrated solution of enzyme. It also consists of deoxyribonucleic acid or DNA, ribonucleic acid or RNA, and ribosomes, which plays a vital role in the protein synthesis in the chloroplast. Moreover, we could say that stroma is like the mitochondrial matrix of plants. The next one would be the thylakoid. Comes from the Greek word thylakos, meaning sac or pouch. It is a single-folded vesicles which serves as a compartment for grana. In grana, it is the site where the light reaction occurs. Each thylakoid is interconnected by the organelle called the stroma lamellae. As I've said earlier, grana is the site for light reaction. But what is light reaction particularly? There are two types or phases of photosynthesis. It will be discussed further later, but just a quick overview. Light reaction is a type of photosynthesis that is light dependent and yields NADPH and ATP as its byproduct. Its site, as what I've said earlier, is in the thylakoid, particularly on the grana. While on the other hand, dark reactions are light independent photosynthesis. It utilizes the NADPH and ATP to drive the production of carbohydrates, and it occurred in the stroma. This is known as the Calvin cycle. But have you ever wondered how about those organisms that doesn't have a chloroplast, like those cyanobacteria or the prokaryotes? Usually, they have this so-called chromatophores in order to harness light and uses it to synthesize their food. As we can see on the figure below, it is a prokaryotes and those circles are their chromatophores.
Acer is a genus of trees and shrubs, commonly known as maples, It has approximately 132 species, most of which are native to Asia, with a number also appearing in Europe, Northern Africa, and North America. Most maples usually have easily identifiable palmate leaves and are most valued and known for their autumn color. This natural phenomenon of changing colors can also be seen in leaf decay. But why does this happen? Why do leaves change color? In this topic, we will be digging deeper on the science of light, pigments, and the roles of both in sustaining life on Earth. Nature has put itself the problem of how to catch in flight light streaming to the Earth and to store the most elusive of all powers in rigid form. The plants take in one form of power, light, and produce another power, chemical difference. This is a quote from Julius Robert von Mayer, a German physician, chemist, and physicist, and one of the founders of thermodynamics. He recognized that plants convert solar energy into chemical energy. However, what exactly is light and how is it turned into energy? The sun emits an enormous amount of electromagnetic radiation, a form of energy that behaves like a wave as it travels through space. Humans can only see a fraction of this energy, which we refer to as visible light. Light behaves both as a wave and as tiny discrete packets of energy called photons. The energy of each photon is directly related to its wavelength. Shorter wavelengths mean higher energy, described by this equation. When a photon hits a molecule, its energy perfectly matches the energy difference between an electron's current ground state and a higher excited state. The photon can be absorbed. This causes an electron to instantly jump to the higher energy level, driven by the molecule's specific structure, particularly systems with many connected double bonds that create precise energy gaps. Once absorbed, this energy can then be used for productive photochemistry, or it can be lost as heat or even re-emitted as light. Centuries of observation led to the understanding that plants use green stuff for growth. It wasn't until 1817 that scientists like Pierre-Joseph Pelletier and Joseph Beneme-Caventou successfully isolated this green pigment which they named chlorophyll. Chlorophyll is a type of pigment that is usually found in plants, and it is what's responsible for plants being of the color green. Photosynthesis is a light-driven process. Therefore, it starts when pigments absorb specific wavelengths of visible light. Organic pigments, whether in the human retina or the chloroplast thylakoid, have a narrow range of energy levels that they can absorb. Energy levels lower than those represented by red light are insufficient to raise an orbital electron to an excited state or quantum state. Energy levels higher than those in blue light will physically tear the molecules apart in a process called bleaching. Our retinal pigments can only see or absorb wavelengths between 700 nanometers and 400 nanometers of light, or the range of visible light. For the same reason, plants' pigment molecules absorb only light in the wavelength range of 400 to 700 nanometers. But if we have our retinas to perceive light and colors, how do plants absorb this light and then use them as their energy source? Going back to chlorophyll, Caventou and Pelletier, along with later researchers, revealed its complex structure with its central magnesium atom and extensive system of double bonds to be crucial for its light-absorbing function. Chlorophylls, especially chlorophyll A and B, feature feature a large ring-like structure called a porphyrin ring, which is the main part that traps light. The magnesium ion at its center is vital for fine-tuning its light absorption properties. A long oily phytol tail anchors the chlorophyll in the cell's membranes. Two of the most common types of chlorophyll is called chlorophyll A and chlorophyll B. Both are similar in structure except chlorophyll A has a methyl group at the C7 position, while chlorophyll B has an aldehyde group at the same position. Both of these two chlorophylls are very effective photoreceptors because they contain a network of alternating single and double bonds, and the orbitals can delocalize, stabilizing the structure. Such delocalized polyenes have very strong absorption bands in the visible regions of the spectrum, allowing the plant to absorb the energy from sunlight. Chlorophyll A plays the central role in photosynthesis as a principal pigment that captures sunlight. Other pigments, like chlorophyll B, are known as accessory pigments wherein they assist chlorophyll A in absorbing the light that it cannot directly absorb. In this case, for chlorophyll A and B, chlorophyll A absorbs light at the range of 430 to 660 nanometers, while chlorophyll B absorbs light at 450 to 650 nanometers. Both reflect a slightly different hue of green, which makes the plant appear green in color. Another pigment also found in plants are called carotenoids. They were discovered by Heinrich Wilhelm Ferdinand Wackenrotter after he extracted them from carrots. Carotenoids have a 40-carbon chain backbone composed of 8 isoprene molecules. Carotenoids are differentiated and produce different pigments by modifying the isoprenoid backbone by cyclization of N groups and oxidation. The amount of pigmentation depends not only on the accumulation of carotenoids but also on the regulation of genes involved in carotenoid synthesis, degradation, and storage. In general, carotenoids absorb wavelengths ranging from 400 to 550 nanometers and reflects yellow, orange, and even red. In photosynthesis, carotenoids dissipate excess energy. When a leaf is exposed to full sun, the light-dependent reactions are required to process an enormous amount of energy. If that energy is not handled properly, it can do significant damage. Therefore, many carotenoids reside in the thylakoid membrane, absorb excess energy, and safely dissipate the energy as heat. These pigments and other proteins form the light harvesting complex, which acts as a funnel to send the light energy into the reaction center. This reaction center then converts light energy into chemical energy through a series of electron transfer reactions. It serves as the core of the light-dependent reactions in photosynthesis, where light energy is absorbed and used to drive the transfer of electrons, ultimately leading to the production of ATP and NADPH, which are used to power the synthesis of sugars. So to answer the question, leaves change color in response to sunlight and the amount of pigments they have. Chlorophyll requires warmer temperature, therefore it is most abundant during the summertime. As the days progress and colder climates come, the chlorophyll production slows down and the existing chlorophyll in the leaves decay. Therefore, the present carotenoids in the plants become visible. Carotenoids also degrade at a much slower rate than chlorophyll. Anthocyanins are uncommonly present in leaves year-round. As the days darken, their synthesis is initiated by increased concentration of sugars in the leaves, combined with sunlight. Their precise role in the leaf is still unclear. There has been some suggestion, however, that they may perform some kind of light protective role, allowing the tree to protect its leaves from light damage and extend the time before they are shed. In terms of their contribution to the color of the autumn leaves, they provide vivid red, purple, and magenta shades. Also not directly related to photosynthesis, but tannins are generally responsible for brown colors and only appear when the leaf is about dead. They are sometimes referred to as phytochemical, which is a non-nutritive plant chemical that has protective or disease-preventive properties. It is a waste product of the tree's metabolism. When the leaf decays, the tannins seep out of the leaves, creating the brown color.
Before discussing the two-center electron transport, let us dwell onto the early discovery about photosynthesis. First, they noticed something strange called the red drop effect. When plants like chlorella are exposed to light of increasing wavelength, let's say from violet to red, they continue to produce oxygen efficiently up to about 675 nanometers. But beyond 680 nanometers, especially in the far red region, oxygen production suddenly drops, even though the plant pigment chlorophyll can absorb that far red light. So this suggested that the light above 680 nanometers alone isn't enough for full photosynthesis. Second, they observe an effect called the enhancement effect. When they shine two different wavelengths of light together, one around 700 nanometers or red light, and another from the yellow-green region, the plant produced more oxygen than the sum of the two lights alone. So this meant that the two lights is working together, not just adding their effects separately. So these two findings led to the discovery that the two photosystems must be working in sequence. This is described in the Z-scheme of the photosynthesis. The two photosystem mechanism of photosynthesis takes place within the thylakoid membrane of the chloroplast, which is a specialized organelle in plant and algal cells dedicated to photosynthesis. The chloroplast is enclosed by a double membrane and contains an internal fluid-filled space called the stroma, where the thylakoid membrane system is suspended. So this membrane system consists of thylakoid discs, which are often stacked into structures known as the grana. So within the thylakoid membrane are embedded the major protein complexes required for the light-dependent reactions, including the PS2 or the photosystem 2, cytochrome B6F, PS1, and ATP synthase, and so on and so forth. Now let's discuss the mechanism how these two photosystems work. So this dual photosystem arrangement known as the Z-scheme is essential because a single photosystem cannot simultaneously oxidize water and at the same time reduce yung NADP+ natin. So the mechanism begins with PS2, which contains a reaction center, which is a special chlorophyll molecule known as the P680. So tinawag siyang P680 because it can absorb light most efficiently at 680 nanometers. So when P680 absorbs a photon or a light energy, it becomes excited, or the P680 asterisk, which can donate an electron sa malapit na electron acceptor molecule called the pheophytin in order to start yung ETC natin. Now, yung P680 becomes P680+, which is a very strong oxidizing agent. And to replace the lost electron, yung PS2 natin, they can oxidize water through this manganese-containing oxygen evolving complex or OEC to split the water into protons, electrons, and oxygen gas as a byproduct. So yung mga electrons, they are transferred to plastokinones, such as the plastokinone A or QA and B or the QB. Then the reduced QB will become PQ or plastoquinol, and this will carry the electrons towards the cytochrome B6F complex. So yung plastoquinone natin is a lipid-soluble quinone molecule, which is a ubiquinone analog sa oxidative phosphorylation natin, characterized by its conjugated ring structure with two carbonyl groups. So in its oxidized form, yung plastoquinone accepts 2 electrons and 2 protons, which reduces the carbonyl groups into hydroxyl groups, forming yung plastoquinol. So as electrons pass through cytochrome B6F, yung protons natin are actively pumped in the thylakoid lumen, contributing to the formation of a proton gradient that later drives yung mga ATP synthesis natin. So the electrons are then transferred to plastocyanin or PC, which is a small copper-containing protein that delivers them to photosystem 1. At photosystem 1, the light is absorbed by P700, which also becomes excited, and it transfers its high-energy electron through a series of acceptors, including yung A sub 0, A sub 1, which is the phyloquinone, and a chain of iron-sulfur clusters Fa, Fb, Fx, until the electrons ultimately reach yung ferredoxinatin or Fd, which transfers them to the enzyme ferredoxin NADP plus reductase or FNR. So, yung FNR naman is ikakatalyze niya si reduction ng NADP plus to NADPH. Now, going back to the goal of photosynthesis, we successfully converted light energy by bombardment of a photon to chemical energy where we produce NADPH. And this is the goal of photosynthesis.
Before discussing one of the light reactions, photophosphorylation, let me define what photophosphorylation is. From the word itself, photo means light, and phosphorylation means an addition of a phosphate group. Chloroplasts generate ATP in much the same way as mitochondria, that is, by coupling the dissipation of a proton gradient to the enzymatic synthesis of ATP. At its core, photophosphorylation is a process by which chloroplasts synthesize ATP. It's the engine that harnesses light energy to drive this crucial synthesis. It's a key part of the light reactions of photosynthesis, essentially converting the energy from sunlight into chemical forms like ATP and NADPH. What's remarkable is that this process shares significant mechanistic similarities with ATP synthesis and mitochondria, a process known as oxidative phosphorylation, both relying on establishing a proton gradient across a membrane, which then powers an enzyme, which is called ATP synthase, to produce ATP. The amount of ATP produced is unaffected by the presence of electron transport inhibitors and observations that photophosphorylation requires an intact thylakoid membrane and that proton translocators uncouple photophosphorylation from light-driven electron transport, provides convincing evidence favoring the chemiosmotic hypothesis, which states that the free energy of electron transport is conserved by pumping hydrogen proton or the proton from the mitochondrial matrix to the intermembrane space to create an electrochemical proton gradient across the inner mitochondrial membrane. The electrochemical potential of this gradient is harnessed to synthesize ATP. This is the general mechanism of coupling of electron transport and ATP synthesis. The proton is pumped out of the mitochondrion by complexes 1, 3, and 4 of the electron transport chain, which is shown as the blue arrows, and thereby generating an electrochemical gradient across the inner mitochondrial membrane. The exergonic return of these protons to the matrix powers the synthesis of ATP, which is shown by the red arrow. It is safe to note that the outer mitochondrial membrane is permeable to small molecules and ions, including the H proton. As we have mentioned the chemiosmotic hypothesis before, we will now explain the evidence or the experimental evidence of this chemiosmotic hypothesis. The strongest evidence for the chemiosmotic mechanism in chloroplasts came from an elegant experiment involving an artificial pH gradient. Researchers soaked chloroplasts in a pH 4 succinic acid solution, allowing the thylakoid lumen to equilibrate to this low pH. When these chloroplasts were then abruptly transferred to a pH 8 buffer containing ADP and inorganic phosphate, they observed an impressive burst of ATP synthesis, about 100 ATP molecules per cytochrome F complex. Critically, this ATP production was unaffected by electron transport inhibitors like PCMU, demonstrating that the pH gradient itself was sufficient to drive ATP synthesis, independent of electron flow. Further observations, such as the requirement for an intact thylakoid membrane and the uncoupling effect of agents like 2,4-dinitrophenol, all provided convincing evidence for the chemiosmotic hypothesis in photophosphorylation. The picture on the left side shows the distribution of photosynthetic protein complexes between the stacked and unstacked regions of the thylakoid membrane. The picture on the right side shows the CF1 lollipops of their ATP synthase project from their stromal surfaces. The enzyme responsible for ATP synthesis in chloroplasts is the CF1-CF0 complex, and it bears a striking resemblance to the mitochondrial F1-F0 ATP synthase. Electron micrographs reveal this characteristic lollipop-shaped structure, projecting from the stromal surfaces of the thylakoid membranes, which are the CF1 units. Structurally, it's composed of a hydrophobic transmembrane CF0 unit that forms the proton channel and a hydrophilic peripheral CF1 unit with a subunit composition, where the beta unit is a reversible AT base. Like its mitochondrial counterpart, chloroplast ATP synthase is inhibited by oligomycin. Functionally, it translocates protons out of the thylakoid lumen into the stroma. This enzyme is strategically located in the unstacked portions of the thylakoid membrane, providing an ample space for the large CF1 globule and ensuring access to ADP in the stroma. The remarkable similarities between chloroplast and mitochondrial ATP synthesis suggest their very early evolution in cellular life. In generating the proton gradient, at optimal light intensities, chloroplasts are capable of generating a significant proton gradient of about 3.5 pH units across their thylakoid membranes. This critical gradient comes from two main sources. First, the splitting of water molecules to produce oxygen releases 4 protons directly into the thylakoid lumen. Second, the transport of the 4 electrons liberated from water through the cytochrome B6F complex actively translocates an estimated 8 additional protons from the stroma into the thylakoid lumen. In total, this means 12 protons are translocated for every molecule of oxygen produced by non-cyclic electron transport. Unlike the inner mitochondrial membrane, the thylakoid membrane is permeable to other ions like magnesium and chloride. The movement of these ions, such as magnesium moving out and chloride moving in, helps to maintain electrical neutrality as protons are pumped. This key difference means that the membrane potential, which is a significant component of the electrochemical gradient in mitochondria, is almost entirely eliminated in chloroplasts. Therefore, the electrochemical gradient driving ATP synthesis in chloroplasts is almost exclusively a result of this large pH gradient. This shows the proton gradient generation. As mentioned, that water molecules produce oxygen and releases 4 protons directly into the thylakoid lumen. Now, let's look at the energetic yield. Most estimates indicate that chloroplast ATP synthase produces 1 ATP molecule for every 3 protons it transports out of the thylakoid lumen. Given that 12 protons are translocated per molecule of O2 produced in non-cyclic electron transport, this translates to about 4 ATP molecules per O2 evolved. However, non-cyclic photophosphorylation also generates NADPH. Each molecule of NADPH has the energetic equivalent of 3 ATPs, adding another 6 ATP equivalents per oxygen produced. So if we consider both ATP and the ATP equivalent from NADPH, the overall energetic efficiency of the non-cyclic process is approximately 1.25 ATP, equivalents per absorbed photon. It's also worth noting that cyclic electron transport is more efficient ATP generator per absorbed photon, yielding two-thirds of an ATP, but it uniquely produces ATP without the accompanying NADPH. To summarize, photophosphorylation is the vital process in chloroplasts that converts light energy into the chemical energy of ATP and NADPH using a chemiosmotic mechanism. This involves the generation of a significant proton gradient driven by both water splitting and the electron transport chain through the cytochrome B6F complex. The chloroplast's ATP synthase, the CF1-CF0 complex, is remarkably similar to the mitochondrial ATP synthase, underscoring its ancient evolutionary origin. A unique aspect in chloroplasts is that the proton motive force is almost entirely due to the pH gradient, as other ions move to maintain electrical neutrality. Ultimately, this efficient ATP generation provides the essential energy for the dark reactions, where carbohydrates and other substances are synthesized from CO2.
So for the last topic under photosynthesis, we're now gonna talk about the Calvin-Benson cycle or the light-independent reactions. So the Calvin cycle is the central process in the photosynthesis where carbon dioxide is converted into organic molecules. It is often called the dark reactions, not because it only occurs in the dark, but because it does not require light directly. However, the Calvin cycle heavily depends on ATP and NADPH that are produced during the light reactions. So this cycle takes place in the stroma of the chloroplast, the fluid surrounding the thylakoids, which provides the energy and organic molecules essential for most life on Earth. So the Calvin cycle was discovered in the 1950s by Melvin Calvin, James Bosham, and Andrew Benson, who used radioactive carbon tracers to carefully follow how carbon moves through this process. So to begin, the Calvin cycle consists of three interconnected phases, which is the carbon fixation, reduction, and the regeneration of ρBP. The cycle is not a one-time process. It is a continuous cycle that both captures atmospheric carbon dioxide and efficiently recycles its starting material to keep going. So this makes the Calvin cycle self-sustaining. So what's essential to remember is that this cycle is responsible for creating sugar precursors that eventually build glucose and other carbohydrates, which are the foundation of most food chains on Earth. So let's now dive into the first phase of the Calvin cycle. So the Calvin cycle begins with carbon fixation, where atmospheric carbon dioxide is incorporated into organic molecules. So the key enzyme in this phase is ribulose 1,5-bisphosphate carboxylase oxygenase or Rubisco, which catalyzes the reaction between one molecule of carbon dioxide and one molecule of ribulose 1,5-bisphosphate, a 5-carbon sugar. So when carbon dioxide binds to ρBP, an stable 6-carbon intermediate is formed, which immediately breaks down into two molecules of 3-phosphoglycerate or 3-PGA. So each containing 3 carbon atoms. So this is the reason why Calvin cycle is also called the C3 cycle. So for every 3 carbon dioxide molecules that enter the cycle, 6 molecules of 3-PGA are produced. So this step is critical because it converts inorganic carbon from the atmosphere into an organic form that can be further processed by plants. So the enzyme phosphoglycerate kinase then catalyzes the phosphorylation of each 3-PGA molecules using ATP, forming 1,3-bisphosphoglycerate or 1,3-BPGA. So this phosphorylation primes the molecules for the subsequent reduction step, effectively adding energy that will later be used to convert them into sugar precursors. So the second phase of the Calvin cycle is the reduction phase, where the energy and reducing power from the light-dependent reactions are used to convert the fixed carbon into a usable sugar form. So in this step, each 1,3-bisphosphoglycerate or 1,3-BPGA molecule is reduced to glyceraldehyde 3-phosphate or G3P, a 3-carbon sugar, through the enzyme glyceraldehyde 3-phosphate dehydrogenase. So this reaction requires an ADPH as source of reducing electrons, and also reduces inorganic phosphate. So for every 6 molecules of G3P produced, only 1 molecule is considered a net gain and can eventually be used to synthesize glucose and other carbohydrates. So the remaining 5 G3P molecule must stay in the cycle to regenerate through BP. So this phase is where the ATP and NADPH produced from the light reactions are directly consumed. Specifically, 6 ATP and 6 NADPH molecules are used to process 3 carbon dioxide molecules entering the cycle. So this reduction step transforms low energy 3-PGA molecules into high energy sugar precursors, moving the cycle closer to producing sugars that the plant can use or store. So the final phase of the Calvin cycle is the regeneration of ribulose 1,5-bisphosphate or RuBP. So the molecule required to keep the cycle turning. So in this complex phase, the remaining 5 molecules of the glyceraldehyde 3-phosphate or G3P are rearranged through the series of enzyme-mediated reactions to form 3 molecules of RuBP. So this step requires additional energy input in the form of 3 more ATP molecules. So these drive the phosphorylation steps necessary to rebuild RuBP. So the regeneration process involves a sequence of molecular transformations including the production of intermediates such as the ribose 5-phosphate, pseudoheptulose 7-phosphate, cellulose 5-phosphate, and others. So specialized enzymes like the transcalotase, ketolase, and aldolase facilitate the transfer of carbon groups between molecules to correctly restructure the carbon skeletons. So without this regeneration, the cycle would halt because there would be no RuBP available to accept new CO2 molecules. So the regeneration phase is crucial for the cyclical continuity of the Calvin cycle and ensures that carbon fixation can proceed repeatedly as long as energy and carbon dioxide are available. So next is the general equation of the Calvin cycle. So the overall net reaction of the Calvin cycle for the production of 1 molecule of G3P is summarized in the equation shown in the screen. So to produce 1 G3P molecules, it requires the input of 3 molecules of carbon dioxide, 9 ATP, and 6 NADPH. So these molecules are used to build the sugar backbone while ATP provides the energy and then NADPH supplies the reducing power. So to make a full glucose molecule, the cycle must turn 6 times, consuming a total of 18 ATP and 12 NADPH. So this demonstrates the significant energy investment required to convert atmospheric carbon dioxide into usable sugars. So the Calvin cycle is carefully regulated to ensure it only operates efficiently when the required energy is available. Light indirectly controls the cycle by increasing the pH and magnesium ion concentration of the stroma, both of which activate key enzymes including rubisco. So additionally, a regulatory protein called thioredoxin plays a crucial role by responding to the chloroplast redox state. So when light is present, thioredoxin activates Calvin cycle enzymes through redox signaling. So this ensures that the cycle runs optimally during daylight. Rubisco's activity is also tightly controlled and is activated under conditions of light and elevated stromal pH. So these regulatory mechanisms prevent the Calvin cycle from operating wastefully in the dark when ATP and NADPH are limited. So this tight regulation protects the plant's energy resources and ensures carbon fixation is synchronized with energy availability. So, photorespiration is a process that competes with the Calvin cycle and reduces photosynthetic efficiency. So, it occurs when rubisco mistakenly binds oxygen instead of carbon dioxide. So, this mistake leads to production of 2-phosphoglycolate, a compound that must be recycled through a costly pathway that consumes ATP and releases carbon dioxide without producing sugars. So photorespiration is especially problematic under hot and dry conditions when plants close their stomata to conserve water, so limiting the availability of internal carbon dioxide. So despite being energetically wasteful, photorespiration may offer protective benefits by dissipating excess energy and reducing the risk of photooxidative damage when CO2 is scarce. So this process highlights the inherent limitations of rubisco and environmental challenges that plants face in balancing carbon fixation and conservation. So, several plants such as corn and sugarcane have evolved an efficient adaptation to minimize photorespiration. So they use a special separation strategy where carbon dioxide is first captured in mesophyll cells by the enzyme PEP or PEP carboxylase, which has a much higher affinity for CO2 than rubisco, and it does not bind with oxygen. So the fixed carbon is converted into 4-carbon compound, typically malate, which is transported to the bundle sheet cells where the Calvin cycle operates. So in these cells, carbon dioxide is released in high concentrations. So this ensures that rubisco primarily binds carbon dioxide and not oxygen. So this adaptation greatly reduces the photorespiration and is highly effective in hot, sunny environments where seafarer plants have photosynthetic advantage over traditional C3 plants. So although this mechanism requires extra ATP, the energy cost is outweighed by the photosynthetic gains and reduced water loss. So for the CAM plant adaptation, CAM or cross-chelation acid metabolism plants such as cacti and succulents have developed a time-based adaptation to survive in extremely dry environments. So instead of separating processes by location, like in the C4 pathway, they separate them by time. So CAM plants open their stomach at night when temperatures are cooler and humidity is higher to absorb carbon dioxide, which is fixed into organic acids like malate and is stored in vacuums. So during the day, when stomata are closed to prevent water loss, the stored CO2 is released from malate and used in the Calvin cycle. So this strategy allows CAM plants to photosynthesize efficiently while conserving water. So this adaptation is particularly beneficial in arid regions where daytime water loss would be detrimental. CAM plants exemplify the remarkable flexibility of plant metabolism in adapting to harsh environmental conditions. So, to summarize, the Calvin cycle is essential in converting carbon dioxide into organic molecules that sustain plant life and, by extension, most life on Earth. So, it consists of three main phases: carbon fixation, reduction, and regeneration. So, they work together to produce sugar precursors while continuously recycling key components. So the cycle is carefully regulated to operate efficiently under light conditions. And although photorespiration can reduce its efficiency, plants have evolved adaptations like C4 and CAM pathways to overcome these limitations. So altogether, the Calvin cycle highlights the remarkable ability of plants to capture atmospheric carbon and turn it into building blocks of life. So that's all for the photosynthesis discussion. Thank you for listening.